Hello everyone, welcome to OK Biology Classroom and welcome to our topic nucleic acids. Today we are going to look at the RNA. In our previous lesson we looked at the structure of the DNA, but today we are going to look at the structure of the RNA, which works so closely with the DNA. But before we get into what we have for today, do well to subscribe to our YouTube channel so that you always have access to our videos whenever they are released. Okay, so remember to subscribe to our YouTube channel. Right. We're going to look at the structure of RNA. In our previous lesson, we were made to understand that within the nucleus, there are these structures that are called chromosomes. And we picked the chromosome and opened up the structure of the chromosome. And when we did that, we found the DNA as an important component of the chromosome. But the DNA isn't the only thing that makes up the chromosome. The DNA, together with some proteins called histones, make up the chromosome. So that is what we were able to grasp from the previous lesson. Okay, but because our focus was on DNA, we picked the DNA alone and we talked about the DNA. And in talking about the DNA, we came to understand that just like Watson and Crick described it as a double helix. So the DNA is made up of two strands, but these two strands have been twisted about each other so that they form the double helix. So we needed to come further down to look at what the main building block of the DNA molecule also is. So we came to talk about the nucleotide, because many nucleotides are joined to form a long chain called the polynucleotide. And a polynucleotide is enough to be a full strand. So that having two strands would give us the full DNA molecule. Right. So we looked at the structure of the nucleotide, which is the building block of the whole DNA molecule. And we found out that the nucleotide has three main parts. There is a phosphate group, there is a 5-carbon sugar, also called the pentose sugar, and there is the nitrogenous base. So we came to understand that the pentose sugar together with the phosphate group make up the backbone of the DNA molecule, or the backbone of the strand. And we also got to understand that the nitrogenous bases that are the inner part of it also are of 4 different forms. So we have the adenine, we have the guanine, we have the cytosine, and then we have the thymine. So we looked at how the pairing is done for one strand being connected to the other, how the pairing is done, obeying the pairing rule as we are supposed to. Okay, but today our focus is going to be on the RNA. So we are saying that at the end of this lesson, the student would be able to describe the structure of RNA, and the student would also be able to identify the parts of the cell in which the RNA works, and as well to be able to distinguish between RNA and DNA. Okay, but the first point I would like you to note is that just like we were made to understand that DNA is deoxyribonucleic acid, RNA also comes from the name ribonucleic acid. So the R is from ribo, N is from nucleic, and then the A is from acid. So RNA, ribonucleic acid. Alright. So now let's move on to the structure. RNA is a single strand. Well, in our previous lesson, we got to understand that the DNA is double-stranded. In other words, the DNA is made up of 2 strands. But it's different with the RNA. RNA has only 1 strand, or the RNA structure is just 1 strand. Okay, but although it's 1 strand, it also is made up of the same building block called the nucleotide. So many nucleotides have been joined to form that single strand called the RNA. So one similarity we find in here is that the RNA also has the same building block as the DNA. So that is the nucleotide. But RNA is single strand. DNA has 2 strands. Okay. Well, RNAs are so important in the body because they are involved so much in protein synthesis. When it comes to forming protein substances within the body, RNAs play so much role in this particular process. So it makes them very important in the body. Right. Because in the body, for example, if the body would have to form an enzyme An enzyme is a typical protein in the body. And when we talk about protein synthesis, it means we are going to join many amino acids to form the protein. Okay, so in forming enzyme per se, the enzyme is going to be formed by joining many amino acids. So that is typical protein synthesis. And RNAs are going to be involved actively in this particular process. But now let us look at how the RNA structure is, because already we have talked about the fact that the RNA is single strand. So like this is how the RNA looks. Now just like the DNA has the pentose sugar and phosphate group as its backbone, the RNA also has same. So the phosphate group and pentose sugar, they form the backbone of the RNA. And the RNA as well has the nitrogenous bases as well. So we see some nitrogenous bases here. So the RNA also has nitrogenous bases. So basically, the building blocks of DNA serve as the same building blocks in here. Although I know we are going to see some differences about them, but when you talk about the building blocks, they are having the same building blocks of nucleotides. So they show the phosphate and then the sugar as their backbone, and as well they have the nitrogenous bases. Okay. But it is important for us to note that RNAs are shorter than DNAs. So we can, in other words, you can say that an RNA is just like a fraction of the length of DNA. Yes. DNA molecule, like in our previous lesson we talked about, DNA molecules are so long, but an RNA molecule is rather short. Okay, so that is one difference that we need to note between the DNA and the RNA. And as well, we also need to appreciate that when we're talking about the structure of the DNA and in relation to the nitrogenous bases, we mentioned that one of the nitrogenous bases of DNA is the thymine. But when it comes to the RNA structure, there is no thymine as a nitrogenous base. Rather, there is a uracil. Okay, so RNAs do not have thymine. RNAs rather have uracil. So that means if you are doing pairing and you're supposed to put a thymine somewhere, if it's where a DNA, you're supposed to put a thymine in there. In this case, that is this an RNA, you're not going to put the thymine in there, but you're going to put uracil there. So RNA has uracil, DNA has thymine. Alright. Now we're going to look at the types of RNAs that we have, and focusing on the well-known types of RNAs that we have. But before that, let us also note this point that in the eukaryotic cell, RNAs are found in both the nucleus and the cytoplasm. Because we are aware that for the eukaryotic cell, the DNA molecule is found mainly in the nucleus. So it is found in the nucleus and it doesn't move out of the nucleus. That is for the DNA found in the nucleus. But RNAs are found both in the nucleus and as well in the cytoplasm. Okay, so what are the types of RNAs that we have? Yes, there are three well-known RNA types. One of them is the messenger RNA. So we usually represent it with a small letter M and capital letters RNA, like we see on the screen. Okay, and then there is transfer RNA, which is tRNA. There is a ribosomal RNA, which is rRNA. So let us quickly look at what the functions of these RNAs are in the cell, starting with the first one that we have on our list here. So we're going to talk about messenger RNA, mRNA. So what does the mRNA do in the cell? Well, the function of the mRNA is to make copies of parts of the DNA. That is its function, to make copy of part of the DNA. The RNA, as we said, is short. It's a shorter molecule as compared to the DNA. So at certain times, there is a need for the cell to make a copy of information on the DNA. So the messenger RNA would be the one to go and make that copy on the DNA and send it into the cytoplasm. So that is its function, to make copies of parts of the DNA and send into the cytoplasm. So we have an image here that is going to help enhance our understanding about how this activity occurs. So we can see that here there is a DNA molecule that has been separated into the separate strands, although not fully. So it has been separated partly here. And then we have the messenger RNAs forming. And note also that RNAs are formed as and when they are needed. So messenger RNAs in this case are being formed because they are needed to pick information from here. So the nucleotides are now going to arrange themselves according to the nitrogenous bases against this part which has been exposed. In fact, they could have done it here, they could have done it here also. So it doesn't really make much difference, right? So in this case, the diagram chose to present them here rather. But as a student, just bear in mind that the copying of the information could be done on either strand. So in this case, it is being copied on this particular strand. So as the nucleotides arrange themselves, then they form a long chain here. So by the time they form this long chain, it means they have fully formed the complement. So whatever information that is found on this particular strand, they have now copied the exact opposite of it, as we know, per the pairing rule. But the opposite of it or the mirror of it doesn't really make much difference. Later you understand why. So they have made a copy of part of the DNA molecule. So that is what we have said here, that the mRNA makes copies of part of the DNA and it sends it into the cytoplasm. That is the work of the mRNA. Okay. So let's move to the next one, the tRNA. Now after the mRNA has made copy of information on part of the DNA and has been sent into the cytoplasm, now the tRNA is also going to copy the information, or is going to use the information on the messenger RNA. So it's like a copy of the copy. That is what the tRNA is going to do. So like we read here, it makes copies of the mRNA. That's it. The only difference that we will be seeing in this particular case is that one tRNA, for its work, it always comes with 3 nucleotides. So one tRNA comes already with 3 nucleotides. Unlike the case where when we saw the messenger RNA being formed, it was single nucleotides that came to do the work of copying. But tRNAs show slight difference by coming with 3 nucleotides each time. But they're going to copy exactly the same thing though. They're going to copy exactly what they have to copy from the messenger RNA. So like we see it. So the yellow part here is showing us the 3 nucleotides that have been joined. So this is one tRNA. So you see that here it is A, but because A is supposed to be joined or paired with T, if it were the DNA, A would have been paired with T. But in RNA there is no T, but rather there is U for uracil. There is uracil. And so instead of a thymine here, it is a uracil. Okay, so another A here connecting to a U, another A here connecting to a U. And then we have another tRNA here. So the G here is connected to the C here. A here is connected to the U here. U here is connected to the A here. And there is another one here. This one is going to be positioned this way. Because there is A here, it means it's going to pair with U here. A here is going to pair with U here. G here is going to pair with C here. So like I said, had this one been the DNA molecule, would have said that A here would pair with T here, then this A here would have paired with T here. But in RNAs, there is nothing like a thymine, rather there is uracil. So A will pair with U here, A will also pair with U here, and G here will pair with C here. So this is how the pairing would be done in the case of the tRNA and the messenger RNA at the time when they would have to transfer information. Alright, so that is the work that the tRNA does in the cell. Okay, so from here, let's move on to the rrna, ribosomal RNA. And the whole process of protein synthesis, we'll be dealing with it into detail in one of our subsequent classes, yes. So don't worry about it if what was being shown on this screen wasn't really making full sense to you. When you get to protein synthesis, you are going to understand everything. It's very easy to understand. We have not got there yet. Here we are talking about the functions of the various RNAs. So just for you to have a feel of what they do, that's why they have been presented here. So just focus on the functions, what they are supposed to do, okay, and not the process and how it is being done as shown in the diagram. Let your mind rather go more on their functions. Okay. Ribosomal RNAs, they are found in the ribosomes, and their main work is to direct the steps involved in the protein synthesis. So talking about how the messenger RNA and the tRNA are going to pair up with each other so that the right information would be copied and that the right proteins would be formed. The rRNAs are going to facilitate that work. Okay, so far this is what we have for RNAs. Yes, we have established that RNAs also have the same building blocks as DNAs, but RNAs are shorter than DNAs. RNAs are also a single strand as compared to DNAs that are double strands. Okay, and we have also said that when you talk about the nitrogenous bases, you come to understand that the RNA does not have thymine, but rather it has uracil. And RNAs work both in the nucleus and the cytoplasm, but DNAs are fixed in the nucleus, especially when you talk about the eukaryotic cell. Okay, so let us get to understand these differences between them, and as well appreciating their similarities. Okay, and we have talked about the various types of the RNAs that we know, or the well-known ones as we call them. And we have talked about the messenger RNA, which copies information from the DNA and then sends the information into the cytoplasm, because DNA is in the nucleus, so it will send the information into the cytoplasm. Then we talked about the transfer RNA, that also makes copy of what has been copied from the DNA. So it copies the information from the messenger RNA. So it is like from DNA, information is copied onto messenger RNA. The messenger RNA also copies its information onto the tRNA. Okay, then the ribosomal RNA, which is the rRNA, will direct the steps involved in the protein synthesis. So that's what we have under RNAs. So in summary, the differences are that RNA is single strand, DNA has two strands. RNA comparatively is short, but DNA comparatively is long. RNA has uracil as a nitrogenous base, but DNA has thymine as one of the nitrogenous bases. RNA in eukaryotes are found in the nucleus and as well in the cytoplasm, but DNAs are found only in the nucleus. And RNAs are temporary structures because they are formed when they are needed. So for example, the messenger RNA is formed when it is needed. But DNAs are permanent structures because they are fixed within the nucleus. And from the name, we get another difference because we know that DNA is deoxy, and deoxy means it is short of one oxygen. Deoxy, less of oxygen. So that makes RNA has one more oxygen as compared to DNA. And so on the other hand, DNA has less oxygen, one less oxygen as compared to RNA. Okay. But for their similarities, we have been made aware that both the DNA and RNA, they make new copies using the nucleotides because they are both formed by joining nucleotides. So that's one similarity between them. And then they both have phosphate group and pentose sugar as their backbone. And also both are involved in protein synthesis. Okay, so this is much easier, right? Okay, so for today's class, in summary, this is what we have talked about. We have looked at the structure of RNA, then we have looked at the types of RNA and their functions: messenger RNA, transfer RNA, ribosomal RNA, the well-known ones. And then we have looked at the differences between RNA and DNA, as well as looking at the similarities between them. Okay, so this is the end of our second lesson under the nucleic acids. In our next lesson under the same topic, we are going to look at DNA replication, or how copies of DNA are made in the cell. Alright, so I have these questions for all of us. The first one says that state 3 types of RNA and their respective functions. And the second question says that tabulate 4 differences between RNA and DNA. If you can't tabulate, you can just state the differences, but make sure you bullet them or you arrange them in points so that they can be easily read. Then the third question says that one strand of DNA shows the following bases: A, G, C, A, T, T, G, C, A, C, C, A, A, T, G, T, C, G. Now, unlike the previous situation in our previous lesson, where we were looking at the complementary code or the complementary information of the nitrogenous base on the DNA molecule, this time we are going to look at the information that the messenger RNA will copy from this DNA. So this is a DNA molecule, A, G, C, A, T, T, up to the last 2 letters C, G. That whole thing is a part of the DNA molecule. So we are going to look at if a messenger RNA is arranged against this particular portion of the DNA, how would the information on the messenger RNA look like? So you are supposed to write them. That is the question number 3. Okay. So this brings us to the end of today's lesson. Thank you so much for being part of our lesson today, and hope to see you in our next class. Okay. But don't forget to subscribe to our YouTube channel.